Hello, my name's Scott Berry. I'm a biostatistician with Berry Consultants. I'm going to talk to you today about adaptive platform trials. It's really a different way of thinking about the world of clinical trials, and I want to describe to you the statistical efficiencies of a common arena for clinical trials. Okay, in order to think about what a platform trial is, let's think about what a traditional trial is. A traditional trial, typically you have a single drug, drug 1, compared in disease A. We ask a single question: does this drug work? Is it safe and effective in this population? We ask single questions. Our trials are straightforward, highly effective for answering that one question. The world has changed a little bit, and we're recognizing the heterogeneity of many types of diseases, where we're worried about the differential benefit or efficacy and safety within subsets of the population. So think about a disease that can be stratified into A through K. These could be ordered. So A through K could be ordered where it's severity. B is more severe than A, C more severe than B, and so on. It could be genetic markers. So these are separate categorical groups. Could be based on demographic, any disease subtype. Could be the timing of the intervention. A is you get in really early. B is slightly later. K is the latest you can get in the intervention. But we're running umbrella trials where we're investigating, does drug 1 work within the strata of the different subsets of disease? We can do innovations within that, dropping subsets, enrichment designs, these types of innovations within umbrella trials. Those are quite common in single pharmaceuticals because they can use their single drug within that. Now, a platform trial extends the clinical trials in the opposite direction, where we investigate many drugs within a disease. And let's think about a single disease at this point, where I can investigate drug 1, 2, and 3 simultaneously in the trial in the same protocol. This isn't separate protocols. This is not a collaborative group. This is a single protocol that investigates multiple drugs. I leave 3 dots at the end of this to represent a perpetual trial. The nice thing about these platforms is they don't necessarily have to start up, run, and stop with the costs and the time involved in that. The trial can run in perpetuity. We add more drugs into the trial, and it can continue on. This is also commonly referred to as a master protocol. Now we can combine those 2 things together. We have multiple drugs, we have multiple strata of disease, and we might even learn as the trial goes on different subsets of the disease. So we can think about those expanding as much as the drugs expanding and this lasting in perpetuity. Now, within a trial like this, where we have multiple subsets of disease, we have multiple drugs, continues in perpetuity, we get great deal of efficiency through adaptation, through innovation at this point. We now have a K by N problem that we used to tackle one at a time. Now we're tackling simultaneously, so we can create a great deal of statistical efficiency with the platform trial design, hence an adaptive platform trial. All right, so I'm going to describe some efficiencies that come out of this. So I'll take a couple generic examples and we'll look at what's the efficiency in these 2 generic examples. Showing you, highlighting what this can look like and the promise of doing things in this common master protocol. So let's take a generic example where drug development typically follows a silo approach where we sequentially test different therapies. Think of each one of these blue boxes as a clinical trial, a 1-to-1 comparison, placebo or control to active agent. We're going to put 100 patients on each one of those in a single trial. If the first experimental agent wins, the process is over and we've got a winning drug. We'll talk about the likelihood it actually works, but we're going to stop the process and we're done. If not, we're going to go to a second trial, a new trial where we do 100 to 100 with agent 2. And then we'll go agent 3 and agent 4, pausing between trials and investigating each therapy. We're going to do a frequentist test, 0.0251 type 1 error in each individual bucket, and we'll continue that process until we get a winner. So what does this look like? One of the really important parts to this that is new as a statistician, we typically talk about alternative scenarios, null scenarios, mixtures of those. We never talk about the universe of therapies. We now have a process that investigates many drugs. So we have to say, what does that look like? Are most drugs effective? Most don't work. The process we cycle through the drugs is critical to understanding the universe of therapies. So I'm going to assume for this simulation that most therapies in this particular situation don't work. That cycling through is a productive aspect of this. So 90% of therapies that we draw out of the universe and stick into our trial don't work. They're the same as the control, which is a 30% success rate on an individual patient. If we pull an effective therapy out of this universe, it has a 50% success rate in any particular patient. We want to find those. We want to find those effective therapies in this universe of therapies. So here's what happens with that universe of therapies in this sequence of trials in 1,000 runs of these processes. Each process has a number of trials. So we're simulating processes now. The average number of trials before we say, aha, we've got an effective therapy is 10.7. We use on average 2,138 patients before we determine an effective therapy or our statistical significant therapy. 1,474 patients failed in the process. 50% of all those patients were placed on a placebo. Now, the percentage of processes that went through and we had a statistically significant therapy that actually found a truly effective therapy was 85%. So our process is pretty good in this scenario of finding effective therapies. 85% of them found an effective therapy. The percentage of effectives that get dumped into the process that are labeled as effective is 80%. 100 to 100 has 80% power for 30% to 50%. So we maintain that, of course. 2.5% type 1 error. Now, because of the integer valued problem here of 100 patients, we actually get a simulated 1.6% type 1 error in this scenario. Okay, so what is the alternative to silo trial after silo trial? We are going to first do a shared control design. It's a very basic platform trial where we're going to run a sequence of trials that have shared control. So we're going to put 5 of the experimental agents, pull 5 of them out of the universe, stick them into a trial and compare to placebo. We're going to do pairwise independent tests. I want to mirror what we did in the old process. 2.5% type 1 error for each one of those. And if any one of those 5 win against a shared control, notice we already are saving 400 patients that we would have used in the old process and stuck on placebo, that now we don't do that because we have a common control. If any one of those 5 are statistically significant, we stop the process and we've got an effective therapy. Sometimes we get 2 of them actually in this process, so a little better in that way. And then that continues on. So what happens when we run 1,000 of these processes together? We get an average of 2.7 trials. Each one of the trials is slightly bigger than the last time. We get an average of 1,600 patients. So by just putting them together in a shared control, the process saves about 25% of patients that need to go into it before we get it. That would be consistent with time, patience. Actually, if you count the white space between them, there's even more savings in time. And that can be extremely big, actually. The average number of, the percent of patients placed on placebo throughout the process is only 16%. 1 out of every 6 on placebo instead of 1 out of every 2, which is part of the efficiencies. We even have the number of processes is more likely to get an effective therapy because we have 5 at a time. So this jumps to 90%. 80% type 1 error. Every agent has the same type 1 error as we did in the single process. So immediately by doing shared control, we save 25% of time, resources, and we're just as strong, and we're treating patients less likely to be on a placebo, which patients certainly like. Now let's think about a perpetual platform trial. We are going to do the same setup where we have a control and 5 agents at a time, but instead of stopping and running a whole separate trial, we're going to pull a therapy out that doesn't work and stick another one in, and it will continue perpetually until we find a statistically significant arm. We are also going to allow an arm to go a little bit bigger if it's looking promising. So we'll do interims every 50 patients for each agent in the trial. At that point, we'll throw it out for futility if it's not doing well. We'll declare it successful if it is successful. Otherwise, it continues with 50 more. So each one is adaptive in its comparison to placebo. We throw it out and bring new ones in, so we've got a single process that goes on. We'll throw a therapy out if the probability has at least a 10% advantage drops below 30%. I'll show you an extra scenario where we add 10%. We want to be more certain it doesn't work before we throw it out. If it shows statistical significance, 98.5% posterior probability with uniform priors, this is like a 1.5% alpha in a frequentist test. Otherwise, we continue until we find an effective therapy. We're not doing response adaptive randomization, which would add more to it. In this case, we're just showing what happens in being perpetual and adaptive within each one. Okay, the third row is for that 30% futility, where we are pretty aggressive in cycling therapies out that aren't looking very good. Notice we have one process here. We don't stop, have white space between them. We have one single trial. The average number of patients is 850 patients. Compared to 2,100, it's less than half the patients, less than 16% on placebo. So again, 1 out of every 6 on placebo instead of 1 out of every 2. The probability a process stops with an effective therapy is very similar to the silo design. It's 86%. So we're doing as well as that. The percent, if an effective enters in the therapy, we are actually more aggressive at making sure that works. 91% chance if an effective enters this process, we show it's successful. We do better on that as well with less than half the patients. Type 1 error is the same. Now, if we want to be more sure it's futile, the 10%, our sample size goes up a couple hundred. And you can vary each one of these in many, many ways. We're showing you 4 designs, 50% savings, increase effectiveness, treating patients better. By the way, we also get a direct comparison between the agents in this, where we don't get it using the traditional method in this setup as well. One more combination platform I'll show you and show you some of the statistical efficiencies of this. Suppose we have 4 agents and we also want to investigate the combinations of these 4. Put them together in 2-way combinations, no bigger than 2-way combinations. So we've got 10 regimens. You can see here we've got 4 single agent regimens and 6 combination regimens for 10 total. We want to do adaptive randomization, favoring the regimens that are doing better for 250 patients. You could see this could go on perpetually, but we'll look at, for statistical efficiencies, what happens at 250. Assume every patient is dichotomous. We're going to do updates every week. We're going to update the adaptive randomization. We enroll 10 patients per week. The first update we'll do at 30 patients and then every 10 patients. We're incorporating an accrual lag of 20 patients. So every adaptation has 20 patients we don't know their data for. So we're assuming there's a 20 patient lag in information time. So after the burn-in of equal randomization for the first 30, we'll do response adaptive randomization where the probability for an arm is proportional to the probability it's the most effective regimen. We adjust that for information, but think about it as the probability it's the most effective. This continues in perpetuity. Now we'll stop it at 250 to evaluate operating characteristics, but you could imagine evaluating it at any time. We're going to use a simple statistical model with coefficients for each factor and then an interaction term for each of the combinations of them with prior distributions for those terms. So 6 combinations, 4 single agents. This is what happens on average under the null. In each of these scenarios, I'm going to show you balanced by the two-way part here, where a 4-4, for example, is just 4 by itself. If you look at 1 and 4, that's when you're using the combination of 1 and 4 as the regimen. The true probability is the blue number in each box. So this being the null, every agent has a 40% probability. The mean number of subjects allocated to that regimen through 250 is the number at the top of each box. The probability it's the winning agent, thought to have the highest posterior probability at the end of 250 patients, is the number at the bottom of the box in this case. And these tend to be very similar probabilities because it's the null. That's the probability of that regimen winning the trial. In the parentheses right next to that in the bottom of the box is the probability if we didn't do response adaptive randomization. So we did fixed probabilities to the 10 regimens through 250 patients. The circles, the green and red circles, show the average sample size, and it's the average number failed is in red, and the total is the average sample size, also shown in black at the top of the box. That it gives you a little bit of flavor for where patients are going in the trial. The average number of failures in the process is shown in the box to the right. So it's 100 here on the null. It doesn't matter what you give, 20% of patients, 40% of patients are going to fail in this process. The nice thing here is we do like a fixed trial. Actually, we do slightly better than a fixed trial because we get away from agents that aren't working early. But it distributes patients evenly. It's looking for something that works. So it works like you'd expect under the null. Scenario 1 is a scenario where agent 4 works really well and the other 3 agents are irrelevant. They have a 40% failure rate whenever 4 is not involved and a 20% failure rate whenever 4 is in there. You can see we distribute many more subjects because of the response adaptive randomization in agent 4's spot. In this case, an average number of subjects of about 50 in each one of these arms. Very high probability of selecting these, very low probability you would select anything else. In this case, the number of failures in the trial is about 10% less. Absolute 20 subject difference for slightly under 10% difference. So this is working more effectively. In a scenario where 4 is even better, we're putting even more patients on 4. Very few patients, an average of 5 to 6 patients on the other arms. We learned very quickly 4 is where it's at, and we're putting huge numbers there. We're decreasing the number of failures by about 35 in this case, with very high probability of selecting 4 as a winning regimen. In scenario 3, notice in the blue truths here that 3-4 is the unique optimal regimen. We're putting an average of 90 of the 250 patients on that regimen. We're selecting it 89% of the time as the optimal way to treat a patient. If we did fixed randomization, that would be 81% with exactly the same model. So the difference here is we put more patients right near that one. We stay away from 1, 2, and 3, and we learn more effectively the best treatment. the same time, we're treating patients better. We have a 20 patient absolute difference by 250 in the number of failures. Again, reducing by about 10%, a little less than 10%, the amount of failures. We're treating patients better. We're more likely to get the right answer in this process. In scenario 4, the 3-4 is even better, and in fact, 1 is detrimental in this case. We put far fewer patients on 1. We put 131 out of 250 on 3-4. We learned very, very quickly that's where it's at. The number of patient difference is almost 20% failure rate reduction by using this particular design. Okay, so what I wanted to show you here was a couple trials that are using this approach. We have Prepare, it's funded by the EU, and they have a platform like this in community-acquired pneumonia and influenza, where they have this process that's investigating therapies with multiple domains of therapy, immunomodulation, antivirals or antibiotics, organ support. So we're pairing these together, so we might have 8 or 16 different regimens, response adaptive randomization. We're treating patients more effectively and learning in the process at the same time. They're also, one of their charges is to be ready for a pandemic outbreak. However unlikely that may seem, we have a trial ready to go immediately. The PCAST report, this comes from the White House itself, that they have a report for the impending issues with antibiotics and the dearth of antibiotics and the worry over drug resistance of the bugs themselves, that we won't have enough antibiotics to handle this. So their report says one of the solutions to this is to develop platform trials for antibiotics, where multiple new agents from different sponsors can all come and play in the same arena. And they reference the I-SPY 2 trial, which is really one of the earliest, maybe the first platform trial that's getting a lot of press as well. So they reference that. This is a solution to the antibiotic problem. IMI knows that the impending problem financially with Alzheimer's, we're not dying of cardiovascular things as much, oncology things as much. We're getting older as a population. Alzheimer's is creating bankrupt situations for governments that pay for the care of them. It's incredibly expensive care, so the governments are saying we need treatments for this. We need to prevent Alzheimer's. So they've created one of their solutions in December of 2013. The IMI put out a call for we need a platform trial. This is the only solution to this. We need a platform trial, and they're going to put 53 million euros towards a solution to this. So we need to develop a platform trial for that. This is being done in animal studies. There's a grant proposal in stroke models to have multiple stroke models, as many as 10 treatments simultaneously, creating 45 combinations to investigate in rats what has neuroprotective capabilities, cycling through very, very quickly. This can be done in early development as much as clinical trials. So in summary, these are very effective. They don't focus on does drug 1 work? It focuses how do we treat this disease? We can do it effectively, we can treat patients better, and oh, by the way, we get right answers more often, more effectively, cheaper, and faster. So the world is changing. You don't have home phones anymore. Cable is going away. You don't get a newspaper anymore. You read things online. These are dinosaurs. The single trial, the single sponsor clinical trial will be next. In 25 years, platform trials will dominate. It has to. They're so incredibly effective. We may look back and laugh about the time when sponsors ran clinical trials. Now they just develop drugs, and we let somebody else be much more effective at running platform clinical trials. Thank you.